Welcome back to Statistics for Genomics. In the last video we took the t-test apart and found 6 ways Genomics breaks it. This video is about something that comes before any test at all. It is about the moment you compute a summary statistic and quietly stop looking at your data. There is a 50-year-old demonstration that makes this impossible to argue with. It is called Anscombe's quartet, and by the end of this video you will never trust a correlation coefficient the same way again. Here is the setup. In 1973, a statistician named Francis Anscombe published 4 small datasets. Each one has 11 points. Each point is just an X and a Y. That is the whole thing. 44 numbers. Nothing complicated, nothing modern, no computing power required. And yet these 4 datasets have been quietly embarrassing analysts for 50 years because of what happens when you summarize them instead of looking at them. Now let's do what everybody does. Let's summarize them. Mean of X, 9.0. In all 4. Variance of X, 11.0. In all 4. Mean of Y, 7.50. In all 4. Variance of Y, 4.12. In all 4. Correlation between X and Y, 0.816. In all 4. Fit a linear regression, and you get Y equals 3.00 plus 0.50x. In all 4. R squared, 0.67. In all 4. Every summary statistic you would normally compute, every number you would normally put in a table, is identical. So they are the same data. Obviously. What else could this mean? Stop here for a second. Because this is the exact position you are in every time you read a method section, or a results table, or a supplementary spreadsheet. You have the summary. You have the correlation. You have the regression. You have R squared. That is what gets reported. That is what gets reviewed. That is, in practice, what gets believed. And you have every reason to think you know what this data looks like. You do not. So let's plot them. Here they are. 4 datasets. Every summary statistic identical to 2 decimal places. And they could not be more different. Dataset 1 is what you expected, a linear relationship with some scatter. Dataset 2 is a perfect curve, a clean parabola, with no noise at all. Dataset 3 is a perfectly straight line with a single outlier that has dragged the regression away from it. Dataset 4 has X fixed at 8 for 10 of the 11 points, and 1 point sitting far away at 19, and that single point is generating the entire correlation. Take a moment with this. Same mean. Same variance. Same correlation. Same regression line. Same R squared. 4 completely different realities. The summary statistics did not fail. They reported exactly what they measure. The problem is that what they measure is not the shape of your data. Let's take them one at a time, because each one is a distinct failure mode you will meet in real data. Dataset 1 is the honest one. A linear relationship with random scatter around it. This is what the correlation of 0.816 is describing, and for this dataset, that description is fair. If all 4 had looked like this, the summary would have been enough. This is the baseline. Everything that follows is a departure from it that the summary cannot see. Dataset 2 is a parabola. Not approximately a parabola. A clean, deterministic curve with essentially no noise. Now think about what the correlation is telling you here. 0.816. That is a strong linear correlation, and it is being computed on data that has no linear component at all in the way you mean it. The relationship between X and Y is perfectly deterministic. If you knew the curve you could predict Y from X exactly. And yet Pearson's R reports 0.816, because Pearson's R only measures linear association. It does not measure whether a relationship exists. It measures how much of the relationship happens to be straight. That is a different question, and the gap between those 2 questions is where a lot of published conclusions live. Dataset 3 is a perfectly straight line, with 1 point off it. 10 of the 11 points sit exactly on a line. 1 does not. And that single point has pulled the fitted regression away from the true relationship, and it has dragged the correlation down from 1.0 to 0.816. So the summary is now describing neither thing. It is not describing the tight line that 10 points are on, and it is not describing the outlier. It is describing a compromise between them that corresponds to nothing real. If you saw the plot, you would immediately ask what happened with that one sample. If you saw only R equals 0.816, you would conclude there is a moderately strong relationship with moderate noise, and you would be wrong about both halves of that sentence. Dataset 4 is the one that should genuinely worry you. Look at the x values. 10 of the 11 points have x equal to exactly 8. There is no variation in x at all. Except for one point, sitting alone at x equals 19. And that one point is generating the entire correlation. All of it. Remove that single observation and there is no relationship left to measure, because x is constant and a constant cannot correlate with anything. So R equals 0.816 is being produced by a single data point. This is leverage, and it is the most dangerous of the four, because in a table it looks exactly like a real finding. And in genomics this happens constantly. One donor, one batch, one sample with an unusual value, quietly generating an association across the whole analysis. So what was Anscombe actually arguing? His paper opens by attacking a belief that was common then and is arguably more common now, that computing is a substitute for looking. He listed the assumptions people held. That numerical calculations are exact but graphs are rough. That for any data set there is one set of calculations constituting a correct analysis. That performing intricate calculations is virtuous, whereas looking at the data is cheating. He was writing in 1973, when making a plot was genuinely expensive and slow. Today a plot costs you one line of code and about 400 milliseconds. The excuse is gone. The behavior has not changed. And if you think 4 datasets is a small demonstration, the idea generalizes much further than Anscombe could show by hand. In 2017, Justin Machka and George Fitzmaurice at Autodesk Research published a paper called Same Stats, Different Graphs. They used simulated annealing to construct 13 datasets that all share the same mean in x and y, the same standard deviation in x and y, and the same Pearson correlation, all agreeing to 2 decimal places. One of them is a star. One is a set of parallel lines. One is a circle. And one, famously, is a dinosaur. The point is not that the dinosaur is funny. The point is that the constraint of matching summary statistics is so weak that you can hit it deliberately with essentially any shape you want. Your summary statistics do not pin down your data. They barely constrain it at all. Now, none of this is news as a curiosity. Everyone has seen the dinosaur. The reason it stays a curiosity is that people file it under fun statistics fact and then go back to reading correlation tables. So let's make it concrete. Here are 6 places in genomics where you are running Anscombe's quartet on yourself, right now, without noticing. Trap 1. Correlation on sparse single-cell data. You want to know whether 2 genes are co-expressed, so you correlate their expression across cells. Sounds reasonable. But a single nucleus matrix is mostly zeros. So what dominates that correlation? The cells where both genes are 0. You are not measuring co-expression. You are largely measuring co-detection, whether the 2 genes happen to be captured in the same cells. 2 genes with no biological relationship will correlate strongly if they have similar detection rates, because they share the same zeros. Plot the joint distribution and you see it immediately, a huge pile at the origin and a thin scatter elsewhere. Compute R and you see 0.6, and you write down co-expressed. Trap 2. This is Anscombe's dataset 4, wearing a lab coat. You correlate a gene's expression against donor age across a cohort. You get a significant correlation. You write down age-associated gene. But plot it, and one donor sits far out on the age axis, and that single donor is generating the entire association. This is exactly dataset 4. 10 points with no spread in X, and 1 point doing all the work. And in an aging cohort this is close to guaranteed, because age distributions are almost never uniform. You have a cluster of donors in one range and a couple of extremes, and the extremes carry the regression. If you have never fit a model against age and not plotted the raw points, you do not know whether you have a trend or you have one donor. Trap 3. Simpson's paradox, which is Anscombe's lesson with an extra twist. You pool your donors and find a gene goes up with disease. Plot it split by batch, and within every single batch it goes down. The pooled trend and the within-group trend point in opposite directions. This is not a paradox in any mystical sense. It happens when your grouping variable is associated with both your predictor and your outcome. In genomics that is the default situation, not the exception, because batch is almost always confounded with something. The summary statistic cannot warn you about this. Only the plot can, and only if you color by batch. If you have ever made a scatter plot and not colored it by your known covariates, you have looked at the plot without actually looking. Trap 4. The beautiful UMAP that means nothing. This one is Anscombe inverted. Instead of a summary hiding the structure, you have a picture manufacturing structure that is not there. A UMAP will always give you islands. Feed it pure noise and it will give you clean, well-separated islands, because that is what the algorithm is built to do. It optimizes local neighborhood preservation and it makes no promise whatsoever about the global arrangement. So the distance between two clusters on your UMAP means nothing. The size of a cluster means nothing. The fact that two clusters look far apart means nothing. And yet those are exactly the things people read off it in figures. So the lesson from Anscombe cuts both ways. Do not trust a number without a picture. And do not trust a picture without knowing what the picture is entitled to say. Trap 5. Fold change without the counts underneath. A log2 fold change of 5 looks enormous. It is the kind of number that goes straight into a figure. But a fold change is a ratio, and a ratio does not tell you what it was a ratio of. 2 counts versus 64 counts gives you a log2 fold change of 5. 2000 versus 64,000 gives you exactly the same number. Those are not the same finding. The first is noise with a good publicist. This is why the EMMA plot exists, and why you should be looking at one every time. It puts the fold change on one axis and the mean expression on the other, so the low-count genes with spectacular ratios are immediately visible as a fan opening up on the left. One plot and the whole problem is obvious. Trap 6. The p-value with no effect size attached. With enough cells, everything is significant. We showed you why in the last video. The standard error shrinks with the square root of n, so at 30,000 cells you can get an astronomically small p-value for a difference in mean expression of 1/100th of a count. Statistically significant. Biologically meaningless. A volcano plot helps here, because it forces the effect size onto one axis, and you can see that your smallest p-values are hugging the center line where the fold changes are tiny. But even the volcano is a summary. It does not show you the distribution behind each point. The gene at the top of your volcano might be beautifully separated between groups, or it might be driven by one donor, and the volcano looks identical either way. So if the lesson is plot your data, the obvious next question is, plot what, exactly? Because plot your data is useless advice on its own. Here are the 5 diagnostic plots that actually catch things, in the order you should run them. Start here, because this is the single most informative plot in all of differential expression, and almost nobody makes it. Take your raw p-values, before any correction, and histogram them. Under the null hypothesis, p-values are uniformly distributed. That is what a p-value is. If nothing is going on, every value between 0 and 1 is equally likely. So the shape of that histogram tells you what happened, before you look at a single gene. A flat histogram means no signal. A flat histogram with a spike near 0 is what a healthy experiment with a real signal looks like. The flat part is the nulls, the spike is your true positives. And then there are the shapes that mean something has gone wrong, and those are the valuable ones. And here are the shapes that mean something is wrong, which is where the real value is. A hill in the middle, with p-values bunching up around the center rather than being flat, usually means your null is wrong. Often that is unmodeled confounding, or a batch effect you did not account for. A histogram that slopes up toward 1. So you have too many large p-values, generally means your test is conservative, something has overcorrected, or your variance is inflated. And a U-shape, with a pile at both ends, usually points at a discreteness problem or a two-sided test on data that will not support it, which for low-count genes is very common. Learn these 4 shapes, and you get a free diagnostic on every differential expression run you ever do. It costs you 1 line of code and it will catch problems that no amount of staring at a results table would ever surface. Plot 2 and plot 3 go together, because they both interrogate the count structure. The EMMA plot puts log fold change against mean expression. What you want to see is a cloud centered on 0, symmetric, with the spread widening as you move to the left where counts are low. What you do not want to see is the whole cloud sitting above or below 0, because that means your normalization has failed and you are looking at a library size difference, not biology. The mean variance plot is the companion. Plot each gene's mean against its variance. You should see variance climbing with the mean. If you fit VOOM, this is literally the trend it is estimating, and looking at it tells you whether the model has anything sensible to work with. Plot 4. Before you cluster a single cell, view a PCA of your samples. Pseudo-bulk each donor, run a PCA on those, and color the points by every piece of metadata you have. Batch, sex, age, sequencing run, processing date. Whoever did the dissection. What you are looking for is simple. Does anything separate on PC1 that is not your biological variable of interest? If your samples split cleanly by batch on the first principal component, you have found the dominant source of variation in your experiment, and it is not biology. That is worth knowing before you spend 3 weeks on downstream analysis, not after. This plot takes about 30 seconds and it has saved more projects than any method I know. And plot 5, plot the gene. The actual gene. Not the fold change, not the p-value, not the position on a volcano. When a gene comes out of your analysis as a hit, plot the raw values, 1 point per donor, split by group. That single plot answers questions the whole pipeline cannot. Is this driven by one donor? Are the groups actually separated or do they overlap almost completely with one outlier pulling the mean? Is the defect in the direction you claimed? You will discard hits at this stage. That is the plot working, not the plot failing. Every top hit in your paper should have been looked at this way, individually, by a human, before it appeared in a figure. So here is the whole thing as a checklist, in the order you should run it. PCA of your pseudobulk samples, colored by metadata, before anything else. Mean variance trend, to see whether your model has something sensible to fit. Emma plot, to check your normalization did what you think. P-value histogram, before you correct anything. And then plot your top hits individually. 5 plots. None of them take more than a minute. Together they will catch the majority of the errors that make it into print. There is a deeper point here, and it is worth being precise about, because plotter data gets repeated so often that it has become a slogan rather than an argument. A summary statistic is a compression. It takes your data and throws almost all of it away, deliberately, and keeps one number. That is not a flaw. That is the entire purpose. The mean is useful precisely because it discards everything except the center. The problem is never that the summary compressed your data. The problem is forgetting that it did. Anscombe's quartet is a demonstration that the compression is lossy in ways that matter, and that the loss is invisible from inside the compressed version. You cannot recover the shape from R. You cannot detect the outlier from the mean. The information is gone, and nothing downstream will tell you it is missing. The plot is not a nicety. 5 things to carry out of this. 1. 4 datasets with identical means, variances, correlation, regression line and R² can look completely different. That is not a trick. That is how weak those constraints are. 2. Pearson's R does not measure whether a relationship exists. It measures how much of it is straight. 3. In genomics, dataset 4 is everywhere. 1 donor, 1 batch, 1 unusual sample, quietly generating an association. 4. The p-value histogram is the cheapest diagnostic you are not running, and its shape tells you whether your null is even right. 5. A summary is a compression, and the loss is invisible from inside it. The plot is your only access to what was discarded. That was video 2 in the Statistics for Genomics series. Next we look at distributions in biology, normal, Poisson, negative binomial and zero inflated, which data generates which and how to tell by looking. Full tutorials, books and code are at multiomeacademy.com. If you want to work through this on your own data, reach out at multiomeacademy.com/contact. Check multiomeacademy.com to learn more.